This segment is on the question of applying information theory to estimating protein probabilities. Is this analysis, is the use of information in the analysis of protein probabilities helpful or hurtful? Now, Professor Dave, it's kind of hard to understand what he was saying regarding this. He was insinuating if you didn't understand information theory, you couldn't argue the probability of information to code for DNA. At least that's the way I heard it. If not, that's not really the point I'm trying to make or address. I'm just trying to say there's no need to go into the topic of information theory. It is something that I studied because I studied electrical engineering and computer science. Your information theory to death in a variety of ways, and I just never see the relevance of applying this discipline to estimating protein probabilities, and I will try to explain why I have that particular viewpoint. Here are multimeric proteins, and I'll explain what multimeric proteins. Most proteins associate into multimeric complexes with specific architectures. That's from a recent article in Nature. A multimeric protein is actually a complex of multiple polypeptides to make a functional system. So actually it takes sometimes more than one copy of a polypeptide. So when the gene expresses, it makes a polypeptide, which we sometimes call a protein. But for the sake of clarity, I'll use the word polypeptide. When the gene is transcribed and then translated, it makes a polypeptide. And sometimes we may need more than one copy of that polypeptide to make a functional protein complex. So here are some examples of those cases. We have these are what we would call multimeric proteins. That's because it is made of the same polypeptide but multiple copies. On the left here is a topoisomerase protein. The blue and the red indicate two identical copies of the topoisomerase polypeptide. This is a homohexameric helicase. Hexameric indicating that there are six identical copies that are expressed in the gene to make one functional helicase complex. This, I believe, is a potassium ion channel. It is a homotetrameric protein, which means there are four identical copies to make this protein. Now, there are heteromeric proteins that have parts from that are expressed in different genes. So if this is the case, this is that most proteins associate into multimeric complexes, then this poses interesting probability problems, and I'll cover that a little bit. This again is a different depiction of a helicase protein that I showed earlier. This is just colored differently, and the different colors indicate there are 6 identical copies. I'll point out that this has hexagonal symmetry. You can see that it approximates a hexagonal symmetry. So there is, it kind of makes sense that we use multiple copies to make something with symmetry like this, because a lot of, we can kind of see the idea of this in the man-made world. There are lots of things in our tools and parts and machines that have symmetry. Symmetry is very useful, is a very useful mechanical concept in making useful devices and parts, such as this hexagonal nut. Another example of symmetry is a pair of scissors here in the man-made world, and for topoisomerase there is that same kind of symmetry, and topoisomerase itself is a pair of scissors. It cuts, it literally is a pair of scissors, but more than that, it cuts DNA in order to untangle it. It then untangles it and then stitches it back together, so it's more than just a pair of scissors. but one can see the value of having multimeric complexes that implement symmetry. Here is another example of symmetry. On the left, in the man-made world, we have these square nuts, and on the right is a ion channel. And maybe just to exaggerate a little bit, I'm going to modify the picture so you could see the symmetry that I'm seeing. See it there? It looks a little bit like a square nut if you look at it from that perspective. So symmetry is very important, and that's just one part of building a multimeric protein. I'm depicting here the topoisomerase 2 in humans, the topoisomerase 2 alpha protein. I'm showing the spelling of the protein in terms of its amino acids, and also some of the structural features. So for those with biochemistry backgrounds, you'll recognize the legend here of the alpha helix. Oh, excuse me. Let me. I had a little bit of a button problem there. One can see the alpha helices here and the beta strands and then the turns. So for those with biochemistry backgrounds, they'll understand this. I got this data from the uniprot.org website, and this is from an this diagram is for an unpublished, still to be published, educational article I'm writing. And you can get this data very easily. It's public domain. You can go to the uniprot.org website, type in top2A, and you'll get a listing of organisms with the topoisomerase 2 alpha homolog. So this is the spelling here, and that's one layer of organization that shows what we call the secondary structures with the color coding of the secondary structures. There is also another way we can depict the same spelling but color coding differently the important functional regions here, and that's why hence a different color coding. Just to give an idea of the complexity and the intricacy of this, if you see here this DQ, when I see that I think of Dairy Queen. A colleague of mine, his graduate work was on figuring out, independently confirming the role of the DQ, and it is important in magnesium binding. So this is a metal binding protein, and it's also ATP binding, and it does all sorts of things. It's amazing that something of only about 1,500 amino acids could have so much functionality in it. So these regions, again, can be mostly identified through the Uniprot website that I referenced earlier. There are papers that might be needed to identify, like this region here. I got that from a paper, and you can look that up if you look the bipartite nuclear localization signal. Hopefully you should be able to find it. But that's not the most important thing. That's just some information. So these, again, are multimeric proteins. You could see the complex architecture and the layers of organization. Not to mention, they need to be able to connect together. And to understand the complexity of the difficulty of creating connectable parts, I'm just going to quote from Bruce Albert's Essential Biology, 5th edition, figure 214. So we have two molecules here. These are proteins, and you could see the charge distribution on them that's caused by the amino acids themselves. And I'll just read the caption. A large molecule, such as a protein, can bind to another protein through non-covalent interactions on the surface of each molecule. In the aqueous environment of a cell, many individual weak interactions could cause the two proteins to recognize each other specifically and form a tight complex. Shown here is a set of electrostatic attractions between complementary positive and negative charges. Now, this is an interesting feature. A lot of times, when we get something, say, from a hardware store or whatever store, and it requires assembly, you have to position the parts correctly and connect it. in the cell, there are systems that will spontaneously assemble in the random motions of the cell. These parts will just kind of jiggle around. They'll find each other and connect, which is a very interesting property. They'll have just the right strength of attraction to find each other and then connect. So that's a marvelous property of proteins that facilitates self-assembly. And perhaps to highlight some of the points here too, I'm just going to show this toy. It's kind of a 3D puzzle. You can the kids would just put all the pieces together to fit just nicely and perfectly. And there's an analogy to this at the protein level. Even for a single protein, like say this EZH2, and I'm sorry it's kind of cut off here, you can't really see that it says EZH2, but that's the name of the protein. This is the amino acid color coding of the regions of the amino acids in the protein. There are about 950 or so amino acids, maybe slightly more. Amino acids in the EZH2 protein, and they color code the regions, and then you can see the color coded regions here. And you can see that there are parts in this diagram. This is what they call a space filling diagram on the right. This is what the protein looks like, a depiction of it with the regions color coded, and you could see how the color coded regions fit nicely and interlock into each other. For the sake of completeness, over here is what they call a Richardson ribbon diagram of that same protein. This is used a lot to help understand the secondary structural characteristics of the protein. So the biochemistry students out there would understand what I mean when I say secondary structures. The EZH2 protein is itself part of a larger complex. Oh, by the way, just let me show this. So this is trying to show kind of the idea kind of in this toy world. You kind of see this toy world where we have very nice precise connections and geometry, and we also have that at the protein level. So what's amazing is the way we spell this changes the shape, and that's actually how the spelling relates to the shape is the protein folding problem. And we can pose this problem to biochemists and other scientists and say, if you wanted to have a protein of a certain shape, how would you spell it? And that would not be a trivial problem. Now the EZH2 complex here, featured here, is actually part of a much larger, the EZH2 protein is part of a much larger complex called the polycomb repression complex, and all these other named bubbles here are proteins, and there are all sorts of things that this connects to. It connects to non-coding DNA, it connects to DNA, it connects to histones. It's an amazing complex. But then the point is we have all these interlocking parts that have to be geometrically shaped in order to connect. So that geometric shape is dictated by the spelling and plus some other factors. But suffice to say, this is a non-trivial problem in making a functional fold. The functional fold means, particularly for proteins that are part of a larger complex such as this, means that we have to have the capacity to interlock with other proteins in a nice usable shape, such as illustrated here by Bruce Alberts. So going back to the topoisomerase here, just to show you give a little bit more details on this problem of connectivity and geometric folding, which informs us a little bit intuitively of the difficulty of forming functional proteins. It gives us the idea of the improbability of this happening from a random sequence. What I did is I took X-ray crystallography data from X-ray crystallography experiments that are publicly posted on the PFAM website, and I used another publicly available software suite called PISA to infer where the connections are. So as in Bruce Albert's diagram here, these charges here are associated with specific amino acids. And so for one protein, we have these specific amino acids. And then on the other protein, we have these specific amino acids with the opposite charge. And in this case, this is a homodimer. These amino acids would basically connect to a copy somewhere else on the other amino acid. It's actually a different coordinate. So this, like say this purple V here in this position probably would connect to a different position on the copy. And this is laid out in the table that's generated by PISA. I haven't had the chance to build the software that will actually help visualize the connections beyond what you're seeing in this diagram. That's kind of a project that's down the line where hopefully one could click on the amino acid here in the listing and you could see where it actually is here and then what the corresponding amino acid might be. So like say an amino acid here might connect to another amino acid here. That's just for illustration. I don't have the listing before me right now. But suffice to say, this one can see that there is a problem from a difficulty standpoint of building something like this from just using random polypeptides. One, it has to be shaped right, as Bruce Alberts ably illustrated with his diagram. It has to be shaped right, and then one has to have the right amino acids with the right charges, corresponding charges, to enable the connection. This is non-trivial. This is non-trivial. And so this would have to have, and this layer of organization for all the connections, that's on top of the other stuff, other layers of organization that I'm pointing out here. So there's a layer of organization in terms of the secondary structure. There's a layer of organization in terms of the functional regions here, and all the connecting regions and the binding regions, et cetera, to help implement the scissors. And then there is another layer of organization that helps the multimeric protein connect to the corresponding parts. This is non-trivial. I'm not giving exact numbers, but it wouldn't be outrageous to say that this is an astronomical problem. Not to mention that if we have a homomultimeric protein as depicted here, as such as those depicted here, it doesn't make sense in a Urey-Miller scenario to try to invoke the emergence of multimeric proteins, because like say the topoisomerase, you'd have to make one polypeptide and then make one that is at least close enough in similarity to that polypeptide to make a homodimeric complex like this. So you need to make two almost identical copies using random mechanisms. That's gonna not work so well. And it's even worse for something with helicase where one needs six copies if it's a homohexameric helicase, or 4 copies with a transmembrane protein, such as the potassium ion channel. And Dr. Tour mentioned the difficulty of forming a potassium ion. Check that. Dr. Tour mentioned the difficulty of having a cell without transmembrane proteins. And potassium ion channels are probably a pretty important transmembrane protein. So this shows the improbability of forming multimeric proteins if the cell does not have a functioning genome. There's no way to store the recipe. So right away, at this level of complexity, it would call into question the viability of a cell not to have a functioning genome. Not to mention, without a functioning genome, how would it recapitulate the recipe of proteins for future generations? So that's 2 problems right there. Now a little bit about probability. I'm just going to zoom out a little bit and talk about just probabilities in general. If we had a deck of cards and we randomly picked out from that deck of cards several cards, and then just threw the cards in front of the porch, you would get a pattern like this. It would be correct to say that this is an improbable pattern, but only in the sense that the pattern is not repeatable again by a random process. Meaning if you took back these cards, put them back in the deck, shuffled the deck, and then grabbed another set of random cards at random and then threw them on the porch, it'd be very unlikely you'd be able to get exactly that pattern. Now, some critics of ID theory will say, well, that's all we're doing is we're trying to just say we can't actually duplicate that pattern. But that's not the real problem. That's not the real improbability in question. There is an improbability as a matter of principles, such as illustrated by this tent of cards. This tent of cards is improbable. The improbability of this tent of cards is improbability from natural expectation. It's improbable as a matter of principle. It's this configuration with the tent is more astonishing than this configuration. This one we just think that's product of random processes. This one would stand out and say, hey, this is not the product of a random process. It's astonishing because of the challenge of constructing it in the geometric sense. I hope you can see that. This can't be a product of random processes. You can't have a tornado pass through a pile of cards and expect this to form. And perhaps making the argument rigorous is tough, but hopefully you can see that. And there are other kinds of we can show other improbable structures. And as a matter of principle, it is improbable. It's not so much that we can't repeat it with random processes. It's the fact that a random process is not expected to create such a geometrically coordinated system. So where am I headed with all this? Did I have to use information theory to show the improbability of these systems? No, we don't need information theory and second law of thermodynamics to give us usable arguments as to why the structure is improbable and would require a human or machine or some sort of intelligence to construct. You don't need that. That would just muddle the issue. I hope that, I hope I'm making the case here. And by way of extension, why would we need information theory to estimate the probabilities of protein architectures? Don't need it. The issue really is, what's the probability of a random process creating sequences that will create this? It's that simple. It's that simple, in my opinion. Now, I have challenged ID proponents because I've studied the information theory issue and tried to apply it to ID because information theory was my background. And at first I was very enthusiastic, and then I got very sour on the idea. And that souring began when I started to ask a very simple probability question. I said, okay, use your information theory, your specified complexity or complex specified information analysis to explain why this configuration of quarters, fair coins, is improbable and why you would infer design. And the amount of gyrations that went through just turned me off. I said, it's not that hard, guys. You can use any number of arguments. You can say that this is a violation of the law of large numbers. It has low multiplicity according to the binomial distribution. Someone used the chi-squared metric. And I said, well, okay, you can use these basic arguments, which actually can be used to supplement information theory. You can start with the basic probability arguments without assuming information theory at all. In fact, the reverse is true. You can kind of build information theory on basic probability arguments, not the other way around. And that's really kind of my beef with trying to invoke information theory. One doesn't need it. So the issue of probability is the geometric connectivity and construction, and it's especially poignant in the protein realm because it involves solving and relating the protein folds to the sequences, and that is a non-trivial problem. It's computationally beyond the reach of our capabilities to compute for non-trivial proteins, non-trivial folds. So again, I'm just showing this picture of connectivity. And so what do I have to say? Why use information theory? Why use information theory at all when more direct and clearer arguments are available? Information theory is used to build man-made communication systems like radios, TVs, modems, compact discs, etc. And that's what I studied in my undergrad before I went to study physics and biology. Does information theory really give that much more insight for the effort expended in trying to apply it to the question of protein probabilities? In my opinion, there's little to no need to invoke information theory or thermodynamics. A random DNA string will generate at best a random polypeptide, assuming it will even create a string that will invite transcription in the first place. Random polypeptides will not make transmembrane proteins, nor the proteins that stitch transmembrane proteins into the membrane. Simple. The math and methods of information theory are mostly superfluous to this simple insight. Almost the same, and I emphasize almost the same, can be said for thermodynamics, but not quite, because thermodynamics has something to say about the feasibility of certain folds. But that's an advanced topic. I mean, that's so advanced that for elementary discussions of probability, we probably don't need to go there. I mean, I'm aware of it, but I would think that the arguments of multimeric proteins and arguments such as I tried to describe by just showing the structures like this and the connections and the layer of organization would probably a little bit be more understandable and accessible. If one wants to go to thermodynamics to talk about protein foldings, that's all well and fine if you understand what you're talking about. The word recipe, it conveys the idea of making a recipe for a protein. It conveys the idea of DNA making a recipe for protein. Dr. Tour used the word recipe in his Abiogenesis lectures for the Discovery Institute. And I really like that word because recipe is a qualitative term that conveys the spirit of the problem of storing the recipe or instructions for making the next generation of cells. Using the word recipe is one way to avoid using the word information and thus avoid arguments over the meaning of information. That's just my preference. If you can do better without confusing your listeners, by all means go for it. But I found if we use words like prescriptive information and information, whatever, it just, I don't know, it never resonated with me, and I have an information theory background. Well, that's all I had for this video, and thank you for joining me. The next video in the series is going to be relatively dry, and I don't blame anyone if I'm not offended if you choose not to watch it, because it is a video that is really a supplement and appendix to the first two videos. It deals with mixing entropy and configurational entropy and some considerations of the problem of purification in abiogenesis. It is for nerds, for sure. So with that, thank you for joining me in this series on Dr. Tour's abiogenesis and my little pointers. And take care, and God bless you.